computer analysis diagnosis and computer vision. He has published extensively more than 200 peer-reviewed journal articles and 200 conference papers. He has given numerous talks all over the world, more than 300, and has received numerous awards. In 2015, just to name few, he was made the Cleveland Business Magazine 40 under 40 list. And both in 2019 and 2020, he was named the Pathologist Power List, that is the list of 100 most inspiring professionals in pathology and laboratory medicine. He has secured more than 50 million in grant funding and also co-founded 2 companies. So he's been very busy. He has been also involved in several sponsor research and industry partnership and medical imaging and with pharmaceutical company. I'm very glad to have Anant present today at our pathology ground round. I wish he would have been here in person, but considering the situation, we're going to have to be happy to have him virtually. Anant, thank you very much for accepting our invitation. Welcome, and please, we're looking forward to your presentation on artificial intelligence and computational pathology as a companion diagnostic implication for precision medicine. Thank you.
Thank you very much, Christina. Real pleasure to do this. Yes, unfortunate that I can't be there in person, but this is the new normal, and so we make it work. So thank you for that very kind introduction. I should just mention, apart from my affiliation to Case Western Reserve, I'm also a research health scientist at the Louis Stokes Cleveland VA, developing and applying these technologies for the betterment of the health of our veteran population. As Christina mentioned, I've got a few academic industry collaborations ongoing, so I should just throw that out there. But my true conflict of interest is the fact that I also have a social media account, and that's my Twitter handle there. So a little bit of shameless advertising here. But if you like what you see over the course of this talk, you'd like to learn more, that's my Twitter handle. If you're on social media and like to give me a follow. So I want to contextualize this talk today by focusing first around cancer, but then we'll give other non-oncologic conditions where AI and computational pathology can play a role. But as mentioned in the title of the talk, the focus really for this talk is less about AI and computational pathology for disease diagnosis, but more from the perspective of it as a companion diagnostic. So sorry, that slide skipped there, but the lifetime probability for a man in the United States being diagnosed with some form of cancer is 1 in 2. For women, it's 1 in 3. And yet, when you look at the total mortality on account of cancer in the US, it's still, these slightly dated statistics, but they still hold in 2020. It's roughly around 600,000 men and women who die because of cancer on an annual basis. Now, this is not to take away focus from the huge mortality on account of cancer, but there clearly is a little bit of a discordance between the diagnostic incidence of cancer and the number of people who are actually dying on account of it. And that discordance, can be explained by a few different factors. One is the fact that we clearly have better screening technologies, better biomarkers that allow us to identify the cancer much earlier. We also have better therapies like immunotherapy now. But the fact is that we are throwing the kitchen sink at a large number of these cancers, treating a lot of what are likely to be indolent cancers extremely aggressively. And nowhere is that better epitomized, than in prostate cancer, where you've got the classic problem of overdiagnosis and overtreatment, where 1 in 6 men in the US will be diagnosed with prostate cancer in his lifetime, but only 1 in 40 will actually die on account of it. Now, most of these cancers tend to be indolent, and yet we end up treating a lot of these cancers very aggressively. And not only are there issues related to side effects and morbidity on account of the disease, but we also are now aware of the significant financial toxicity that overtreatment and overdiagnosis can have. And we've seen I'm sure all of you have seen statistics like this, where up to 42% of new cancer patients lose their life savings, many of them within 1 year of the initial diagnosis of cancer. And so in our group, we've really been thinking around AI and computational pathology, not solely from a diagnostic perspective, which is identifying the presence of disease, but really thinking about it from a prognostic perspective, so being able to predict disease outcome and progression, and also from a predictive standpoint. How can we think of computational pathology and AI as a companion diagnostic tool to be able to predict benefit and response to drugs and therapies? And ultimately, it's of course the development and application of these tools that will allow us to truly realize the precision medicine. In other words, using tools like AI and computational pathology for making sure there's an alignment between a patient's risk profile with the appropriate management and treatment strategy. Now, this is not to say that there aren't tools out there to address this need. I'm sure everybody in the audience is aware of Oncotype DX, formerly from Genomic Health, now part of Exact Biosciences. That's a 21 gene expression assay that's used to identify which patients, which early stage ER-positive breast cancer patients can derive added benefit from adjuvant chemotherapy. We know that the vast majority of early stage ER-positive breast cancer patients will do fine with hormonal therapy and surgery alone, but a small subset of them, maybe 20 to 30%, probably need the adjuvant chemotherapy. The 21 gene expression assay from Oncotype, which is in the NCCN guidelines, of course, has really changed the landscape of how we approach treatment for early stage breast cancers, but it comes with a caveat. This is a destructive test. It's a tissue destructive test. It involves physical shipping of the tissue to specialized facilities, and it's also expensive. It's over $4,000 per patient to apply this test. But there's also the issue of intratumoral heterogeneity. We know that more aggressive tumors tend to have multiple subclones. We know that the more aggressive tumors tend to have extreme amount of heterogeneity, molecular, but also morphologic heterogeneity. And so one of the challenges with a lot of these genomic-based tests or molecular-based tests is that they're really dependent on where the tumor is sampled from. And so if you're not really sampling the most aggressive component of the tumor, then the risk profile or the risk score that one obtains may not really be a reflection of the true risk profile for that patient. And therefore, the treatment therapy that's prescribed may not be appropriate. Now, our group, like a few others, have been looking at the role of what we call pathomics or computational pathology, specifically features that can be derived using artificial intelligence, machine learning, image processing, computer vision from routinely acquired tissue slide images to be able to, like I said, not just diagnose the presence of disease, but also be able to prognosticate disease outcome and also to be able to predict therapeutic response. What you see here is a very classical H&E slide, digital tissue slide image that has then been interrogated by computational tools developed by our group. You can see here that the algorithms go in, identify the individual cancer nuclei and lymphocytes. We're able to create graph-based algorithms using computer science and network theory to look at the spatial architecture and the spatial arrangement of these cells, which then provides a series of quantitative attributes, quantitative features that allow us to talk about the spatial architecture in a very quantitative and reproducible way. But we can do other things. We can start to look at the textural parameters and the textural heterogeneity within the different tissue compartments, within the individual nuclei, within the stroma, within the epithelium, and convert this entire whole slide pathology image into highly quantitative, reproducible features or a digital biomarker that now can be associated with outcome as well as therapeutic response. So my journey in the space of AI and computational pathology started about 15 years ago when I was working with Mike Feldman and John Tomaszewski, then at the University of Pennsylvania. Mike Feldman is still at Penn and the vice chair of pathology, and really got exposed to this area of whole slide images and digital pathology. And so we started to work together to develop algorithms for grading of breast cancer, automated machine-based grading of prostate cancer. Published a number of papers in the late 2000s around this area. But around 2007, 2008, I had the good fortune of meeting Sridhar Ganesan, a breast oncologist at Rutgers Cancer Institute of New Jersey. And Sridhar looked at the work I was doing and said, you know, the grading that you're doing for breast cancer and prostate cancer using algorithms is fantastic. But the fact is that as an oncologist, that's not really my pain point. What I want to know is how to manage the patient. How do you treat the patient? And so could you pivot a little bit? Could you think about thinking of these tools as grading and decision support tools for the pathologist, and think about it as decision support tools for me as a clinician? In other words, can you look to connect features from these pathology images with aggressiveness and outcome for the patients directly, and see whether we could stand up an image-based risk score, very similar to the oncotype risk score, but one that looks at patterns from pathology images, with the goal being that we can now risk stratify patients based on the patterns mined from the images, so that the high-risk patients could be the ones, or might be the ones that might benefit from adjuvant chemotherapy, whereas the ones with the lower risk profile might be the ones who could be spared adjuvant chemotherapy. So how do we go about developing these kind of risk scores, these image-based risk scores or IBRIS algorithms? Well, I think everybody probably in pathology, but I would dare say, you know, anyone working pretty much in any field has probably encountered or heard of deep learning. Deep learning has become ubiquitous in so many different fields. Of course, it's not a new concept. The idea of neural networks has been around for over 50 years. But what really has happened over the last 10 to 15 years is that with increase in computational horsepower, we've been able to create much more sophisticated algorithms where we can create layer upon layer upon layer of neural networks, stack them together and create what are called deep learning architectures for machine learning. And the beauty of these algorithms is that they're largely unsupervised. In other words, all you need are a stack of images or training images. You need the annotation of the target of interest. Providing that to the network then is able to learn patterns in an unsupervised way to distinguish the target of interest from everything else. So you see, this is work we published about 5 years ago, where we trained a network using annotations of nuclei from H&E images, and the algorithm was able to output the exact location of the individual nuclei. Now, this, of course, has been taken and applied to multiple different areas for series of different applications, including driverless navigation. We took this approach. We were one of the first to apply this approach in the context of computational pathology. We put out a tutorial/review paper about 4 years ago, where we demonstrated that beyond just individual cancer nuclei, these approaches could be used to identify a series of different kinds of primitives: epithelium, stroma, tubules, mitotic figures. More recently, in fact, just a month ago, this paper came out in Kidney International. We're working with Dr. Laura Parassoni at Duke. We've taken these approaches and applied it to finding a series of different primitives in kidney pathology. In fact, we applied it to finding glomeruli, interstitial capillaries, proximal or distal tubules from multi-stained kidney biopsies. So not just H&E, but looking at PS stained and silver stained and trichrome stained images. So multiple implications of these approaches for detection of individual primitives, structures in pathology images, and for segmentation. One of the big questions with these approaches is the amount of effort required to create the training sets, because at the end of the day, these algorithms are only as good as the data that's provided to them. And towards that end, you need detailed annotations of the structures of the primitives of interest on the pathology images. And that's laborious. That's backbreaking work. And as the saying goes, the term I've learned from my pathology friends and colleagues, there's not sufficient butt time, right, to go in and do detailed annotations of all of these structures and all of these primitives. So how do you address this issue of limited butt time? Well, we've created something called Quick Annotator. It's a tool that allows for interactive annotation of pathology images to be able to create the discovery sets or the annotated learning sets for training these neural network architectures by using just a few candidate segmentations from a pathologist expert. You can stand up some of these networks. The network then results in an output. That output can then be edited by the human reader. And by going iteratively back and forth between the human editing and the network, we're able to stand up pretty detailed annotated data sets that can then help further train and enrich these neural network algorithms. Sorry, just skipped over that slide. This is just another quick example of how these algorithms can work in a fairly high throughput, efficient way. This is some work from our own group, where you see a high resolution 40,000 pixel by 40,000 pixel H&E image. The algorithms go in and very rapidly are able to identify hundreds of thousands of nuclei, lymphocytes on these pathology images. So these tools are really, really powerful, and really from a segmentation classification perspective allow us to identify these structures or primitives of interest very efficiently. So a few years ago, we asked the question, we're looking at the role of deep learning and neural networks for segmentation and detection. Well, could we potentially challenge these approaches to do something at a higher level? And so one of the use cases we decided to look at was in the context of heart failure. And so working with Mike Feldman at Penn, along with Ken Margilis, a cardiologist at the University of Pennsylvania, we decided to pull together a data set of patients where we had access to the endomyocardial biopsies. And so the question we were asking was, could we train a neural network algorithm based on the endomyocardial biopsies and now figure out whether these patients have evidence of heart failure or not? So we did it in sort of the classical way. You take a training set, a discovery set of 100 patients. Patients were equally distributed between those who had heart failure versus those who did not. That was used to train the neural network, and then the neural network was applied to an independent test set of another 100 patients. And so we ran this procedure through. The algorithm came back with a 97% accuracy and area under the curve in being able to predict which patients had heart failure versus which patients did not. We also, for good measure, had two pathologists look at the same test set and laboriously go through all of those images. And now we asked them to figure out whether they could discriminate between patients with heart failure and those who did not. And turned out that when we did a head-to-head comparison, the pathologist had an average area under the curve of 74%. So contrast that with the 97%. So there was a 23% differential, suggesting that the neural network algorithms have really done an outstanding job in being able to predict the presence of heart failure. So, then we got very excited. We put out a press release. Press release got a lot of visibility. GE Reports scored it one of the 5 most exciting stories on the planet for the week. I was briefly trending on Reddit, which is a social media site. So we got a lot of visibility and press. But what is interesting is a couple of months later, we decided to then further extend the validation set. So we requested again from the same site, University of Pennsylvania, an additional 90 patients. And this is the same site, the same lab, the same scanner that was used for digitization of the slides. And these were then sent over, and we ran the network again. And what was amazing was that the network spectacularly imploded on these new cases. In other words, we went from that 97% area on the curve down to 75%. So we were still on par with the way the pathologists were performing, but it was a significant decrease in the accuracy of the classifier from 97% down to 75%. My students spent a lot of time really trying to figure out what happened, because this was not validation where you were going to a completely external site where there were obviously going to be differences in slide preparation, there were differences in pre-analytic factors, batch effects. This was the same lab, this was the same site, the same lab, the same scanner. So what happened? Well, after a lot of brainstorming, and thanks to my amazingly brilliant trainees, they were able to figure out that what had happened between the first batch, the first cohort, and the second cohort was that a software upgrade had been applied to the digital slide scanner. Now that software upgrade, that remote software upgrade, didn't actually change the images perceptually for the pathologist. They could really see no difference. But it clearly had perturbed the images sufficiently for the networks to be confused. So that very, very minor change, almost unanticipated change, had thrown off the networks because of the sensitivity of the slide scanning process. And so that really brings home a very critical message, which really segues into sort of the next part of my talk, which is that we have to be very, very careful of these brute force algorithms. And trusting these brute force algorithms too much is a faith misplaced. Interestingly enough, later this evening, in fact, I'm going to be on a panel discussion, and one of the other panelists is a professor of machine learning named Cynthia Rudin at Duke University, and I have huge respect for Cynthia. And she put out a very, very influential paper last year where she said you have to stop trusting the black box. And so one of the challenges with these neural network algorithms is the fact that there is a huge amount of opacity with respect to what is happening on the inside. These algorithms produce an output, but it's really not clear what they're learning and where and how they arrive at the conclusions that they do. And so Cynthia put out this great paper talking about how, particularly when we're making high stakes decisions, these deep learning black box approaches really should be avoided, because unless you know what's going on within the box, when the box breaks down, you're not going to be able to figure out how to address the issue, how to fix the issue. And so another classic example of this is an anecdote I came across online when I was researching and trying to figure out what happened with our heart failure prediction model. And I came across this anecdote from Samir Singh, faculty member at University of California, Irvine, where Samir had been working with a student to stand up a neural network to try to distinguish huskies from wolves. So now all of you will appreciate that this is a tough problem. Huskies and wolves look very, very similar. And so what Samir had his student do was to go download a bunch of images from Google, train a network based of annotated examples, what was a husky, what was a wolf, train the network, and then the network was run on a set of independent images, very much like what we did with the heart failure patients. And so the network did an amazing job. It had a near perfect accuracy, something like 99%. And Samir, who's clearly way smarter than I am, immediately appreciated that that was not possible. Right? He found out that, he realized this is a really difficult problem. It's not possible for the network to come up with a near perfect prediction. So he and a student went about interrogating what had happened. And the conclusion they came to was that the network wasn't really learning features of the face of the animal. The breakthrough, by chance, what the network had learned was that in all the images involving huskies, there was some white in the background. Right? So invariably there was snow in the background. And what it was learning was that in the images with huskies, there's white. And so it looked for that and really was doing sort of a background classification problem versus an animal classification problem. And so that again really speaks to this issue of not knowing what the black box is learning. And that opacity is a challenge because, like I said, when things break down, because one is not able to understand, appreciate what's happening inside the box, you've got a challenge of how to address this issue. How do you fix this issue if you don't know how the box is actually working? Now, one of the things that we've been looking as an alternative to these black box approaches are what are called handcrafted features, which we believe provide an intuitive alternative to the neural network strategies. Now, I've talked previously about how these neural networks can be used to identify individual cells, individual nuclei. One of the things that these approaches are very useful for doing are also to be able to distinguish different cell types. We can actually distinguish individual lymphocytes from individual cancer nuclei, from macrophages, from fibroblasts. And so it allows for a deep phenotyping of the different cell types within these pathology images. one of the really exciting advances in neural networks, particularly with respect to pathology, is so-called GANs, or generative adversarial networks, that allow for identifying the individual cell types on these pathology images. But beyond that, we can also start to distinguish different tissue types. We can distinguish the stromal regions from the epithelial regions. And so this allows us to not just phenotype the individual cells, but phenotype the cells within individual tissue compartments. And so by doing this, by using the black box or the deep learning as a segmentation tool, as a tool for detection and classification of the individual cells and the tissue compartments, we can now use this processed image as a way of doing biomarker discovery, as a way of extracting and prizing out features that are more interpretable, more intuitive, and that relate more to the pathobiology of the disease. So what I'd like to do for the rest of my talk is to go through some very specific examples of how we're looking at these handcrafted features or engineered features as a way of connecting these patterns to disease outcome, disease prognosis, as well as therapeutic response. So I want to start with some work that I'm very, very excited about, that we've been pursuing over the last year and a half, which is to try to look at collagen patterns, collagen fiber patterns on pathology images. So specifically, we've known for a long time that collagen fibers are implicated in prognosis of multiple different types of cancers. But there's really been no way of quantifying this, of capturing quantitative features relating to collagen fibers. And Haojie Lee, a graduate student in my group, has developed approaches that now allow us from HD images to be able to first segment out the collagen fibers, identify the epithelial regions, this is in the context of breast cancer, and then assign vectors or orientations to the individual collagen fibers, and look at the orientation of the collagen fiber with respect to the boundary of the tumor. So looking within the tumor-associated stroma, but looking at the orientation with respect to the boundary of the tumor. And so what we've done is to come up with a set of features that capture the disorder in the collagen fiber orientation. What we found is that when you look at patients who have short-term survivals, patients who typically tend to do poorly, the collagen fiber orientations tend to be more harmonized, tend to be less chaotically arranged. But in patients who typically tend to do much better, there is more chaotic arrangement of the collagen fibers. And this sort of resonates with what we know from the literature, because these collagen fibers provide the highway for metastasis of the cancer cells, that having a more disordered collagen orientation impedes the quick migration and progression of the cancer cells from the primary site to other sites in the body. And so we've done a study here with ECOG Actrin 2197. This is a clinical trial data set from about 25 years ago, which was looking at ER-positive breast cancers and looking at the role of endocrine therapy and chemotherapy. So we worked with ECOG to get access of the individual tissue slides, and we were able to demonstrate that when you looked at the collagen fiber orientation, that the collagen fiber orientation was an independent prognostic marker of survival in ECOG 2197, and also from slides from the TCGA. So this is one example of a handcrafted engineered feature that obviously resonates with what we know about the pathobiology of the disease. Now, I know, I'm sure a whole number of breast pathologists on the call, I would hope so. And one of the things I mentioned at the start of my talk was the work we started to do with breast cancer grading, and published a number of papers in the late 2000s, really focused on how we could go in and find nuclear pleomorphism, how we could capture tubules, how we could look at mitotic count. And what we've done more recently has been to create a predictor that uses deep learning to first go and quantify nuclear morphology, nuclear pleomorphism, arrangement of the nuclei, quantifies the number of tubules on the pathology images, quantifies the number of mitotic figures on the pathology images, and then creates a multivariable risk score based off patterns that pathologists have known for a long time to be critical in stratifying the aggressiveness of the disease. So again, very intuitive, interpretable features, because this risk score is really coming about based on features that we know are tightly anchored to the pathobiology of the disease. So just some data to share with you. This is now, again, ECOG 2197, but also a cohort of patients from the Tata Cancer Center in Mumbai in India. And you can see here very nicely that not only is this prognostic in lymph node negative breast cancers, but it's also prognostic of survival in lymph node positive breast cancers. So this multivariable risk score is prognostic across both lymph node negative and lymph node positive, ER positive breast cancers. So why is all this important? I talked about the Oncotype DX up front, and this is a little bit tongue-in-cheek, so you'll indulge me a little bit here. So, why is this important? Well, one is the cost factor. You talk about Oncotype DX and other related molecular diagnostic tests. These can be very expensive. For instance, the Oncotype is over $4,000. But if you think of a digital test that only requires digital slide images and access to the algorithms, this could be done for log orders lower in cost compared to the molecular diagnostic test. A very, very rapid turnaround, because with a cloud-based architecture, you could turn around the analysis very quickly. There's no physical shipping of tissue. There's no destruction of the tissue. And therefore, you could potentially have a global footprint, as we're starting to see now with our collaboration with the Tata Cancer Center in Mumbai in India. And so in other words, you have an inexpensive, fast, reliable, accessible prognostic breast cancer test that's priceless. For everything else, there's MasterCard. So sorry, that's my little humor for a Thursday morning. What could we do to potentially improve the molecular diagnostic test? We know the Oncotype DX is here to stay. And so we did a study a couple of years ago, presented this at ASCO, where we went back to the ECOG 2197. We combined the IBRIS predictor with the Oncotype DX score and found that we could actually improve the performance of the Oncotype DX by something like 20%. So again, just to contextualize this, that meant the use of the IBRIS assay, the image features, improved the ability of the Oncotype DX to identify 20% truly low-risk breast cancer patients who could actually successfully avoid adjuvant chemotherapy. So very tangible improvement in the Oncotype DX assay with the use of the image-based classifier. But it's not just ER-positive breast cancer. A study we put out last year in breast cancer research showed that the utility extended to stage 0 breast cancers as well, specifically in the context of ductal carcinoma in situ. So working with Sunil Badve at Indiana University, we were able to create a predictor looking at the shape, the orientation, the arrangement of individual nuclei in DCIS, and connected that with the risk of progression of DCIS to invasive ductal carcinoma. So in other words, we were able to demonstrate that these morphological features from H&E alone were associated with the risk of progression of DCIS to invasive ductal carcinoma. I want to talk to an area that I know is of interest to my host this morning, Christina. In fact, this is work that we've done with Christina and other groups, where we've been looking at trying to predict post-surgical recurrence in prostate cancer. And so this was a fairly large study we did with 6 different sites, almost 1,000 patients. And instead of nuclei, we focused on glands. So trained a neural network algorithm to go and find the glands, extracted a series of features relating to the shape of the glands, the arrangement of the glands, the texture appearance of the glands, and showed that these features from H&E images alone were strongly prognostic of recurrence post-surgery in these prostate cancer patients. And not only was it prognostic in all comers, it was specifically prognostic and really added value in margin negative patients. And so as you can imagine, that was really very, very satisfying, really compelling, because we were able to identify a subset of patients who typically otherwise might have been identified as relatively low risk. We were able to identify a subset of patients who actually tended to have worse outcome and therefore might be candidates for adjuvant therapy of some sort. So this was sort of a very, very welcome finding. Now, one additional thing that we did in the study was go head-to-head with the Decipher test. So I talked about the Oncotype DX test. The Decipher test is a gene expression-based test for prostate cancer and is in the NCCN guidelines for predicting risk of metastasis in prostate cancer patients. Again, it's tissue destructive, it's a molecular test. And so we said, well, can we go head to head? We had a cohort of 173 patients where we had both the Decipher as well as the digital slide images. And so we said, let's see how they do in terms of predicting recurrence. And so what was really nice was that both of the tests were prognostic. So in a head-to-head comparison, the hazard ratios were more or less the same. But then we did one additional thing. We said, let's take the image features and let's drop in just a couple of other standard parameters. Let's just drop in the Gleason grade and let's drop in the preoperative PSA. And so when we did that, what happened was we were able to achieve a hazard ratio that was almost twice the Decipher test. So significant improvement in the combined classifier, the image features coupled with preoperative PSA and Gleason grade, significantly outperforming the Decipher test in the ability to predict recurrence post-radical prostatectomy. So this was a very satisfying result, again suggesting that there's independent information, very, very valuable prognostic information that can be gleaned from H&E images alone. More recently, again, this is work with Christina and a few others. This work is currently under review, where we've looked at quantifying Cribriform patterns. And so we've shown that, again, using deep learning to quantify the Cribriform patterns, we've been able to come up with an index, a quantitative index that captures the Cribriform extent. And that index, that reproducible quantitative index, is strongly associated with risk of recurrence post-radical prostatectomy. While I'm not showing it on this slide, I can tell you that one of the other things that we discovered in the study was that the real value tends to come out in the Gleason grade group 2 patients. So these are the 3+4 patients. And that's really where the Cribriform index really shines. We're a really, really strong C index upwards of 0.8 and seems to really add additional prognostic value in that 3+4 patient population. A paper we just put out recently, this was last month in the journal Cancers, we looked at baseline biopsies. This work with Jonathan Epstein's group and folks at Johns Hopkins, where we looked at a series of features relating to cancer nuclei from baseline biopsies in prostate cancer patients on active surveillance. Pretty old cohort from the 80s at Johns Hopkins. This is part of sort of the initial active surveillance set from Hopkins. We had about 190 patients, used about 60 patients for training the classifier to predict which of these patients on active surveillance would go on to have upgrading or upstaging. Validated this on an independent set of 131 patients and showed that the predictor was associated with risk of upgrading or upstaging in these patients. All of this was done for baseline biopsy. We went head-to-head with Pro-PSA, and the classifier was significantly outperforming Pro-PSA in the ability to predict risk of upgrading and upstaging. One of the other areas we've been looking at is bladder cancer. Again, very, very nuclear-focused tumor. One of the things that we've also looked at in bladder cancers, and again, I'm preaching to the choir here, so I'm sure a lot of you are aware of this, is that the polarity, the orientation of cancer nuclei tends to have a lot of importance in bladder cancer. And so we've come up with a way of quantifying or digitalizing that biomarker. So we've been able to assign a vector to the orientation of the individual nuclei. Again, looked at the entropy or the disorder with respect to the orientation of those individual nuclei, and trained a classifier based off these nuclear orientation pleomorphism related features. So quantitative features from these nuclei then feed into the classifier, and we've demonstrated that not only can we go head to head in terms of bladder cancer grading, as well as pathologists do, but that these features are also independently prognostic of outcome in these patients. Another area we've been working on with Jim Lewis down at Vanderbilt is in the area of head and neck cancer, specifically looking at predicting disease-specific survival in oral cavity squamous cell carcinoma patients. Again, looking at the features relating to the diversity in shape and appearance of the individual nuclei, shown that these features are prognostic of survival and outperform T-stage and end-stage. In a subset analysis, we actually looked at patients who had the AJCC 8th edition staging criteria, specifically looking at stage 1 or the low risk group. Small set of patients, no doubt, but what was really intriguing was that in this subset, we found a distinct set of patients who had worse outcome. And this is really critical because I talked about predictive companion diagnostics. And this is where we move from prognosis into that predictive space, because what this data is telling us is that even though, because of the AJCC 8th edition staging criteria, all these patients were classified as stage 1 and therefore would probably receive surgery alone, our classifier is saying there's a subset of patients who probably should be offered adjuvant radiotherapy. These are patients who probably won't benefit from surgery alone and therefore should be getting additional, more aggressive therapy in the form of radiation therapy. So, implications not just for prognosis, but more critically for modulating therapeutic regimens. Working again with Jim Lewis and David Adelstein and Shlomo Koishman at the Cleveland Clinic, we've now started to look at other primitives. We know that in P16 positive oropharyngeal squamous cell carcinomas, multinucleation has been suggested to be an important hallmark of prognosis. The challenge is being able to visually identify these primitives or these structures. Very, very recently, we've trained an algorithm to go in and do this automatically. Again, a pretty large study with over 1,200 patients. This paper is currently under review. But we were able to demonstrate that just going in and identifying these multinucleation events and creating an index, just like with the Cribriform, created an index for multinucleation, that multinucleation index was prognostic of outcome in P16 oropharyngeal squamous cell cancers. And we've also done a subset analysis within AJCC stage 1, stage 2, and stage 3. And what was interesting was that in stage 1 and stage 3, the subsets, this multinucleation feature was still independently prognostic, even within those individual staging criteria. I think probably by far and away the number one question I get from pathologists is, do I have a way of quantifying tumor infiltrating lymphocytes? And I have to say that is the number one question I get asked. And so we've been actually working on machine learning for TILs, back when maybe it wasn't as hot. We put out one of the first papers on machine learning for identifying TILs in pathology images about 11 years ago. More recently, we've started to look at not just the density, not just the number of TILs, but specifically looking at the spatial architecture of TILs. And so this is a study we put out in Clinical Cancer Research last year, where looking at early stage lung cancer, we used our machine learning algorithms to go in and identify TILs. You can see these green dots here, and specifically identify clusters of TILs. We also trained another machine learning algorithm to go in and identify cancer nuclei. So you can see the red dots and the red clusters here. And then using graph network algorithms, looked at the co-arrangement and the interplay between the cancer nuclei and the tumor infiltrating lymphocytes. And it turns out that the spatial architecture was quite different. So if you look at the patients without recurrence, you see that the cancer nuclear clusters tend to be surrounded by pretty large dominant TIL clusters. On the other hand, when you look at patients who do have recurrence, who have worse outcome, the cancer nuclear cluster tends to be larger, more dominant, and the TIL clusters around it tend to be smaller, more fragmented, not really providing that what one can call a mothering effect or an enveloping effect, allowing the cancer nuclear cluster to really break out and progress out. And that's reflected in the Kaplan-Meier curves as well. So we're seeing that the spatial arrangement, in fact, turned out to be more prognostic than just the absolute number of TILs in the pathology in these H&E images. And so these are all H&E images, by the way. The colors are really generated by the algorithms in terms of the classification of these individual cells. And so that's why it looks like a immunostained image, but it's really an H&E image. Well, what else can you do with that? So beyond showing prognosis in early stage breast cancer, early stage lung cancers, more recently we've shown that this spatial TIL feature is also predictive of response to checkpoint inhibitors in immunotherapy treated non-small cell lung cancers, and also prognostic of survival in immunotherapy treated non-small cell lung cancers. This paper is currently under review. but we put out a few abstracts around this work and shown that across 3 different agents, Nivo, Atezo, as well as pembrolizumab, that these features are predictive response, but also prognostic of overall survival. One of the questions I get is, well, what are you picking up with these TILs? You know, it's all great to go in and find the TILs, but we know that, you know, the TIL composition could be quite myriad. You could have CD4, CD8s, CD20s. So what exactly are you capturing? So working with Kurt Schalper and David Grimm at Yale, we've done studies now where we've taken H&E images, we've taken the corresponding quantitative immunofluorescence images, and co-registered those two. So we've done our TIL analysis on the H&E images, co-registered that back to the QIF images, and looked at trying to understand the molecular composition of these TIL patterns we're identifying on the H&E images. And so this is work that is just about to be submitted for publication, but I wanted to share it with you. Some very, very intriguing findings. What we're finding is that if you look in the context of lung cancer at both adenos as well as squamous cell carcinomas, the TIL patterns that we're identifying that are prognostic of outcome are quite different. So in terms of the squamous cell cancers, it turns out the spatial architecture, spatial arrangement is really important. In adenocarcinomas, on the other hand, it turns out the actual density and the number of TILs seems to be more important. When you look at the QIF images, it turns out, again, the molecular composition of the adenocarcinomas and the squamous cell carcinomas tends to be quite different. You actually see that there is a plurality of CD4s in the adenocarcinomas, but on the other hand, in the squamous cell cancers, you see really a combination of both CD4s and CD8 cells. So what is nice here is that you're not just looking at the morphology, but also trying to connect it to the molecular underpinning of the tumor and really trying to get at sort of the molecular composition of these tumor infiltrating lymphocytes. This is sort of related work where one of the other things that we've done is looking at an unsupervised clustering of the TILs on the H&E images and try to identify whether there are clusters of TILs that are extremely prognostic. So one thing is to consider all the TILs on the H&E image, but we've said, okay, let's do a cluster analysis on the H&Es and identifying distinct clusters, subsets of TILs that seem to be more prognostic. And so when we look again with the QIF, we've been able to map the molecular composition of these prognostically defined clusters on the H&E images. And again, sort of gets back to this idea of trying to really get at the molecular underpinning and the molecular composition of these features that we're identifying as prognostic on the H&E images. A few other cancers that we're looking at currently, this is in the context of ovarian cancer, where the spatial arrangement of TILs has been shown again to be prognostic. This is work we put out earlier this year at the SBI Medical Imaging meeting. What was nice here was we showed that not only was this feature prognostic in ovarian cancer, but it was also associated with prediction of response to platinum chemotherapy. We've subsequently expanded this work and demonstrated both in endometrial cancers and cervical cancers that the spatial TIL arrangement, the spatial architecture of TILs is strongly prognostic of survival in these 2 other gynecologic cancers. We talked about immunotherapy in the context of lung cancer. We've now expanded this out to gynecologic cancers. Just recently at the ASCO meeting, we showed that the spatial arrangement of TILs in 49 patients treated with immunotherapy, this is cervical, ovarian, and endometrial, that these features were strongly prognostic of survival and predictive of response to checkpoint inhibitors. Just another quick example, I talked about multinucleation in the context of P16 positive oropharyngeal cancers. Well, it turns out that TILs are also important. So again, spatial arrangement of TILs, this is in almost 1,000 patients across 6 different sites. We've demonstrated that the spatial arrangement of TILs is also prognostic of overall survival and event-free survival in the context of P16 positive oropharyngeal squamous cell cancers. I started the talk with an example in the context of predicting heart failure from endomyocardial biopsy, showed that with a black box algorithm, we could get about 97% accuracy, and then also showed how, when you had a new set of cases, that accuracy dropped to 75%. We've taken the lymphocyte approach and applied it to biopsies of the heart, specifically to try to predict rejection rate. And what's been really interesting is that we're now looking at the spatial arrangement of myocytes and lymphocytes in these biopsies, and actually gotten to a performance that's slightly better than the deep learning algorithm. We're getting to about a 98% AUC in distinguishing high rejection grade from low rejection grade. We've actually now expanded this out to something like 2,300 patients from 4 different sites, and the algorithm holds. And again, the point I'd like to make is not so much the accuracy, it's just that when you go with intuitive, interpretable features, you have a better shot at generalizability, you've got a better shot at achieving robustness across multiple different sites, and at the same time getting a pretty high accuracy. Part of it is doing for us as computational engineers to do the heavy lifting, to work closely with pathologists, to understand the pathobiology of the disease, and really develop algorithms together that really take advantage of the tremendous domain expertise that you have. And so working in tandem, working together, we can develop algorithms that are interpretable, intuitive, but also generalizable and highly accurate. Just a few other examples as I wind up here. This is in the context of triple-negative breast cancers, where again, the spatial TIL signature has been shown to be prognostic of survival. I have to have a slide on COVID-19. Could not talk about COVID. This is work that's currently under review, where we got a set of autopsy patients working with Sanjay Mukhopadhyay at the Cleveland Clinic. Got a set of images from autopsy patients, patients who died because of COVID-19 as well as H1N1, applied our analysis of lymphocytes to the images from these patients who died because of the 2 different flu infections, and found very distinct immune patterns, that the immune patterns we were seeing in H1N1 tends to show larger aggregates of lymphocytes compared to the COVID-19. Couple of other things I just want to briefly touch on. The other question is, where do you interrogate? So a lot of what I've shown so far has been really focused on the tumor. The question is, should we always only be focusing on the tumor? And so we put out a paper 3 years ago where we looked at regions outside the tumor. We looked at patterns of nuclear morphology, nuclear architecture in tumor-adjacent benign regions, and showed that the features from these tumor-adjacent benign regions were strongly prognostic of biochemical recurrence. In fact, more so compared to the features that we got from within the tumor. One of the standard questions I always get when I show this data is, how can you really be sure that that region you were looking at was normal or benign? Well, my response is always the same. We did this work with Johns Hopkins. Jonathan Epstein identified these regions as normal and cancer. If you've got a problem with whether this is normal or not, take it up with Jonathan Epstein. So, sorry, that's again, just no offense meant there to Jonathan Epstein. It's just a little bit of tongue-in-cheek humor there. So one of the other things we've also been looking at is trying to understand, are there differences in patterns across different populations? So as you build these AI models, these computational pathology models, are there differences across African Americans and Caucasian Americans? We put out a paper in Clinical Cancer Research in April, where we found that there were significant differences in the appearance of stromal nuclei in surgical specimens of African American patients with prostate cancer versus Caucasian American patients with prostate cancer. Why is this important? Well, a lot of the models we currently have for risk stratification, the nomograms, the Catan, the Capra, and so on, don't really explicitly account for population-specific differences. And what we found is that you don't account for population-specific differences, the ability to prognosticate outcome in African American patients was really poor. But when you created population-specific models that invoked explicitly the features that were unique to the disease phenotype in the African American population, then that model did extremely well in prognosticating risk of recurrence in African American patients. So in other words, it seems to suggest that accounting for population-specific disease phenotypes, accounting for those differences, is probably really, really critical, and that we might need to think about creating population-specific risk models for these various different diseases. And it's not just in prostate cancer. We just had a paper accepted, we just had an abstract accepted last night, in fact, in the context of breast cancer, where we did a study where we looked at tissue images from early stage ER-positive breast cancers from women in India, again, working with the Tata Cancer Center in Mumbai with respect to patients in North America, and found there were significant differences in the appearance of the nuclear morphology and the disease phenotype, and that these risk prediction models, again, benefited by taking a population-specific approach. Just a few other examples, just very, very quickly here. This is another area beyond solid tumors. We've also started to look at non-solid tumors. This is in context of bile duct brushings and cytology, where we've been able to develop classifiers to distinguish adenocarcinoma on bile duct brushings. One of the big questions always is the stability and the generalized ability of the classifiers. It's an area that we paid a lot of attention to, not just focusing on accuracy, like I said, but focusing on stability and generalizability. A lot of our studies really focus on multi-site evaluation, multi-site assessment. And so we're always constantly looking to see whether the features are not just accurate and prognostic, but are they generalizable as well. And finally, one of the big questions is the quality of the images, because we all know garbage in and equals garbage out. And so to address that issue, Andrew Janowick, a research faculty member in our group, developed something called HistoQC, which is an automated quality control program for digital pathology slide images. Completely free program. You can, that's the link for the program. You can go in, download it, apply it to your pathology slide images. And the nice thing is it can actually identify a series of different artifacts, issues with tissue folding, issues with pen marks, hair on the glass slide, cracks in the glass, etc., etc. So a series of different artifacts can be identified and could be used to do a quality assessment, quality control of the digital slide images. I don't have time to talk about this, but this is some really fascinating work coming out of the University of Washington. Jonathan Liu, we're working together to now go beyond 2D pathology to 3D pathology, where we're starting to look at, biopsies through something that he's developed called light sheet microscopy, which allows us to look at morphologic features of the disease in 3 dimensions versus just 2 dimensions. All right, so let me end here. So hopefully what I've conveyed is that computational analytics with routine imaging can help address questions in precision medicine. One of the big things about this is that we're talking about routinely acquired data. And so the low cost component is a major driver, I think, in having global impact, particularly in low and middle income countries. Hopefully what I've also conveyed is the importance of an intuitive basis for the approaches and for the features. You can't trust the black box. You really have to think about interpretable features ultimately to drive clinical adoption. It takes a village to do the work, and I'm really just the conductor of an amazing symphony that I get to conduct on a daily basis. In other words, have real privilege and honor of working with an amazing team of trainees, students, postdocs, and faculty here in Cleveland, Case Western. And of course, you always have to thank the people who put food on the table. So very, very fortunate and privileged to have been the recipient and the beneficiary of federal funding. And with that, I will end. Thank you very much.
Thank you, Anant, for a fantastic presentation. And I'm sure there's plenty of questions on the chat. This was very informative and out of the ordinary, of course. Adam, do you want to read the question, or how do we want to proceed? Or you want me to read them?
You can read them since you're already talking, that's fine.
All right, so recent trend, so this is from Dr. Reddy. Recent trend is more towards small needle biopsies instead of traditional open biopsies, like prostate may be an exception, and non-tumor heterogeneity and varying stromal background inflammation or cell response. How AI will help work contribute given the above scenario of the, I guess, variability and
yeah, no, I think that's a great question. So I think that one of the things that you obviously have to put out there is that at the end of the day, these algorithms, these approaches are only as good as the data that's coming in. The core biopsies is something that we work quite extensively with. The data that I showed with immunotherapy response for lung cancer is all based on biopsies. So that's a real small amount of tissue. But we've been able to try to, find features that are associated with outcome and prognosis. Now, we have a partnership right now with a pharmaceutical company where they sent us their clinical trial data sets. There are 500 patients, biopsies from 500 lung cancer patients. We ran our HistoQC tool and found that about 140 of them, the tissue was so small that we couldn't do anything with it. So, one of the qualifications in all of this is, of course, you've got to have a certain minimal amount of tissue to be able to, do the analysis. And I think that's true just for pathologists as well. Right? I mean, you've got to have tissue on the slide in order to render a diagnosis. If there's no tissue, then well, you can't render a diagnosis. But I think that the key thing here is that the amount of real estate needed is pretty small. You don't necessarily, I showed examples both from biopsies as well as surgical specimens. And the nice thing is that if you've got a minimal amount of real estate on the tissue, you can pull features out that you can then connect to outcome and response.
The next one is, in your models, have you used any third-dimensional methods for viewing potential cancer spots? And I think this probably you address a little bit at the end, right?
That's right. Yeah. So that's the exciting work with Jonathan Liu. Amazing collaboration. We just received a DoD grant where we're looking at this three-dimensional attribute, the z-axis, to try to find differences in the phenotype of prostate cancer between African-American patients and Caucasian American patients. So it's hopefully will provide a lens to try to understand morphological differences in 3 dimensions between different patient populations.
This is from me actually. Can most computer extracted feature be generalized to different tumor subtypes or are unique to specific subtypes?
Yeah, so I think it depends. One thing that I showed the one example with the tumor-infiltrating lymphocytes and showed multiple different examples where, the spatial tail features, worked from everything from COVID. It was important for COVID. It was important for cardiac rejection grading. It was important for ovarian cancer, head and neck. But, I think that we want to step away from, trying to come up with that silver bullet that addresses all the different domains and all the different areas. I think the point I would hopefully try to convey through the talk is that there's the real importance of working closely between the computational engineers and pathologists. And we got to work closely together. I've been really privileged and the beneficiary of working closely with my pathology colleagues, learned significantly from them. So this whole, I find this entire conversation or argument so ludicrous about, putting pathologists out of business. You've got a tremendous amount of brain trust, amount of experience, amount of knowledge in understanding the pathobiology of the disease and the morphology of the disease. The way I look at it is, what can we then take from that and try to model computationally so that we can really, embed those domain-inspired features, those domain-specific features that you all know so well from the pathology into these algorithms. Some of these features can have implications in a pan-cancer kind of way, in a pan-disease kind of way, but some of them may be very specific. We know tumor budding. I didn't talk about tumor budding. We're doing some work in tumor budding. Now, that's specific to certain solid tumors. Multinucleation, again, may be specific to certain tumors, but not to others. We know that glands are going to be very important when it comes to prostate, but maybe not across the board for all diseases. So I think, again, the way I see that question, Christina, is that the onus is on the bioengineers, is on the computational scientists to do the heavy lifting, to understand what makes sense for the particular problem you're trying to solve, rather than saying, well, we're going to build a model that's going to address every cancer, and that's all you need. You need one box, and it'll tell you exactly what you need to know. I think that's foolhardy.
I'm sure these will make all the pathology field much better now. So, can the model also cluster the signals of ISH RNA probes and IHC done together?
Yeah, the RNA probes, I don't know. We don't have any data to report. IHC, yes, absolutely. I showed a little bit of the data we've generated with Laura Barisoni, where we've looked at PAS silver stains and trichrome images along with H&E. And we've done work in prostate as well, where we've looked at H&E and NF-kappa B, for instance. So we've done a lot of work with H&E and IHC. I think, again, the critical thing I come back to is, understanding what value each of these different stains and different markers provides. I think that is a really critical question. The reason I always start with the H&E is because there's a lot there, right? Morphology, there's a lot of information, and it's the lowest common denominator. If you can pull a lot of information from that, then that gives you platform to build off, right? And then you can now say, well, if I add in the IHC, what else, what kind of complementary information can I get from those other stains, those other markers? And so that's the approach. So the short answer is yes, you can, but I think, again, the longer, more complicated answer is the critical piece is to try to understand what complementary features can you get from those other, IHC markers, those IHC images.
So now we are out of time. Just the last question is, what is your experience regarding acceptance of AI by pathologists and clinicians?
It's a great question. I think what I've tried to do is, hopefully, is try to demystify the black box. Hopefully what I've conveyed here is that this doesn't happen in isolation. I mean, I have a primary appointment in biomedical engineering. I have a secondary appointment in pathology. I'm very proud of that. I work closely with pathologists. The greatest compliment I can get is when somebody mistakes me for a pathologist. I'm not a pathologist, but I work closely with pathologists. And I think I see this really developing as decision support tools for the pathologist. Unfortunately, the popular press and the media and people in the machine learning literature, make some outrageous statements. You don't need to train pathologists anymore. You don't need to train radiologists anymore. Look, I mean, we've had AI for a long time. I mean, one of the examples I go back to, we put out a paper in Nature Reviews Clinical Oncology last year around AI and digital pathology. I deliberately started that paper by tracing the history of image analysis in pathology and traced it back to a group from Sweden back in the early 60s and the late 50s, but they were doing image analysis of slide images. They were doing snapshots of slides and doing some very rudimentary image analysis back in the 60s. So I think we need to get away from this whole, going to put pathologists out of business, clinicians out of business. I think the opportunity is to work together, collaborate together, and use the complementarity of the different fields and build decision support tools to get beyond the mundane, right? There are things that I'm sure all of you would appreciate. If we had a way that you didn't have to count mitotic figures, I'm sure all of you would appreciate that. That stuff makes your eyes bleed. I have done that. I've seen it. I know that is. Why would you want to do that? Why not use the tools to help simplify some of these low level operations, and then think of how you can really truly add value working in consort. So a lot of it is conversation, discussion, and really demystifying the process. I think once you do that, then you get better synergy and collaboration.
Well, thank you very much, Anant. We wish we would have you for more hours, for more questions. This was excellent. We really appreciate your presentation, Rafi.
Thank you. Thank you.
And looking forward to have you visit in person when things get better.
Absolutely.
Thank you very much.
Thank you so much for having me. Take care. Cheers.
Ciao.
Bye.
Say hi to Cleveland.
Will do. Will do.